Welcome to BioBits and Bytes.
So we're diving into one of the really fundamental challenges in biology today, aren't we?
We absolutely are. It's this question of proteins.
Yeah,
You start with this, well, essentially a long string of beads, the amino acids.
just a one-dimensional sequence.
Exactly. And somehow that string folds itself up, usually all on its own.
Spontaneously, yeah, into this incredibly precise, complex, three-dimensional shape.
And it's not just any shape, is it? It's the shape. The one specific structure it needs to do its job.
Right. It's like trying to predict the final perfect knot in a super long, complex string just by knowing the order of the beads.
That's a good analogy. But with proteins, that final knot, that shape, it's not just about looking intricate. It literally dictates what the protein does.
Absolutely. That shape determines, how an enzyme grabs onto its target molecule, or how a structural protein builds tissues, or how an antibody recognizes a virus.
So if the shape is even slightly off.
Function's usually gone, or at least impaired. And that's often the root cause of many diseases, right? A misfolded protein.
Which brings us to the core problem. Predicting that 3D structure just from the 1D sequence of amino acids. The sequence to structure problem. It's been this, grand challenge for decades. A computational nightmare.
Oh, completely. I mean, the sheer number of ways a protein could theoretically fold is astronomical, just mind-bogglingly vast.
Bigger than the number of atoms in the universe for even medium-sized protein, I've heard.
Easily. So sifting through all those possibilities to find the single correct lowest energy fold, that's incredibly difficult computationally.
For a long time, the best bet was kind of like looking for a cheat sheet, wasn't it?
Yeah, homology modeling.
Yeah.
If your protein had a cousin, a similar sequence whose structure was already solved experimentally and put in the PDB, the protein data bank.
You could use that known structure as a template.
Yeah.
Sort of adapt the blueprint.
Exactly. And that worked pretty well for a lot of proteins. But what about the ones with no known relatives, the unique ones?
The novel folds, or the hard targets, as they call them, where there's no blueprint to copy.
Right. Predicting those structures from scratch, or ab initio, was historically much, much harder. Especially for bigger proteins, the accuracy just wasn't there.
Until recently, that is. The whole field's been turned upside down, really, hasn't it? Mostly thanks to deep learning.
That's the revolution. These powerful AI methods, trained on just vast amounts of sequence and structure data, they somehow learn to spot the patterns.
Patterns that tell them which amino acids, even if they're far apart on the string, are likely to end up close together in the final folded structure.
Exactly. They learned the hidden rules of the protein origami, you could say.
And that context is perfect for what we're diving into today. We're looking at a newer method that really leans into this, integrating these cutting-edge AI approaches with more established techniques. It's called D-I-TASSER.
Right. The DI signaling that deep learning integration, it builds on a long history of the I-TASSER software suite. But this is a significant evolution.
So our deep dive today is all about unpacking this D-I-TASSER method. We've got sources, research papers basically, that describe how it works, how well it performs.
And importantly, how it stacks up against the current heavyweights in the field, like AlphaFold 2.
Our mission, then, is to break all this down for you. Get under the hood of this hybrid approach, understand the key concepts, see the performance data, and try to figure out what it all means for biology and medicine moving forward.
It's this really interesting mix, isn't it? Learning from data with AI, and then using those learnings to guide simulations that are more grounded in physics.
Yeah. Have you ever wondered why predicting protein structure is such a huge deal? Or how computers are actually making inroads on this incredibly tough puzzle? Let's unpack this.
So as you said, it starts simple, a string of amino acids, but function needs that 3D shape, and the sequence holds the key, but unlocking it computationally, that's the beast.
It's not just neighbors interacting, is it? It's amino acids way down the chain that might end up touching or being very close in the final structure.
Exactly. Predicting all those long-range interactions and how they all work together to constrain the shape for a chain that could be hundreds, even thousands of units long. That's where the computational complexity just explodes.
The origami analogy works well here, too. It's not just one fold after another. Folds interact, right? A tiny change early on could lead to a completely different final shape.
And the protein could potentially get stuck in all sorts of intermediate non-functional shapes along the way. Finding that one stable, correct fold is the challenge.
Okay, so historically you had homology modeling relying on known templates, and then the ab initio, from scratch methods, which struggled with size and complexity.
But deep learning changed the game. Training these neural networks on all the available sequence and structure data let them predict those crucial long-range interactions with way better accuracy than before. That's what powered breakthroughs like AlphaFold 2.
That's a great question. Let's break it down into how methods like D-I-TASSER try to solve this. Because it's not just deep learning, is it?
No, exactly. The sources describe D-I-TASSER as a pipeline, a series of steps. And it's explicitly designed as a hybrid system.
So it's taking the power of deep learning predictions
and feeding them into or integrating them with more traditional simulation methods, methods that try to model the actual physics of protein folding.
And the sources make a point of highlighting its specific strategies for handling multi-domain proteins. Those have always been a real headache, right?
Oh, definitely. Multi-domain proteins are like several complex parts that need to fold individually and fit together correctly. Getting that interdomain arrangement right is tough.
Okay, so let's walk through the pipeline. What's step one?
It often starts with getting a really good multiple sequence alignment, an MSA.
Right, the MSA. So you take your protein sequence and you find potentially thousands of related sequences from other species.
Exactly, and you line them all up so the corresponding amino acids are in the same columns.
And why is that so important?
Coevolution. If two amino acids may be far apart in the sequence, consistently mutate together across evolutionary history in that alignment.
It suggests they're probably interacting or touching in the final 3D structure. They have to change in sync to maintain that interaction.
Precisely. That coevolutionary signal is like gold dust for predicting contacts.
So D-I-TASSER uses a tool called Deep MSA2 for this.
Yes, and the sources say it's quite powerful. It uses multiple search tools like HHBlitz, JackAimer.
To search through these absolutely enormous sequence databases.
And critically, not just the standard genome databases like UniProt, it also digs into huge metagenome databases.
And metagenomics, that's sequencing DNA from environmental samples, right?
Yeah.
Like soil or seawater.
Exactly. It gives you access to protein sequences from potentially millions of microbes we can't even culture in the lab.
And the sources say this really boosts the diversity and depth of the MSAs.
Significantly. More diverse sequences means stronger, clearer co-evolutionary signals, which helps everything downstream.
Interesting. And Deep MSA2 doesn't just make one MSA.
No, it actually generates several using slightly different strategies and databases because, one approach might work better for a particular protein family than another.
Okay, so you have a few candidate MSAs. How do you choose the best one? That sounds clever.
It is quite neat. They basically take each candidate MSA and run it through a simplified, quick version of AlphaFold 2, just the core modeling part.
Like a quick quality check.
Yeah. They see which MSA produces an initial model structure with the highest confidence score, according to AlphaFold's own metric, PLDDT. The MSA that leads to the most confident-looking structure gets selected.
That makes sense. Use a known good predictor to vet your input.
And they also calculate a metric called NEF, the number of effective sequences. It's a way to quantify the diversity in the chosen MSA. Generally, a higher NEF means more information content.
Okay, step one. Get the best possible MSA, rich with evolutionary information.
Okay.
Well, step two, finding structural clues.
Exactly, finding clues from structures we already know. This involves a tool called LaMETS3.
LaMETS3. That's a meta-threading server. What's threading?
Threading is where you take your protein sequence and try to, thread it onto the known 3D structures in the PDB library. You see how well your sequence fits onto each known fold.
And a meta server just combines results from lots of different threading programs.
Right. LaMETS3 integrates 11 of them, apparently. Things like HHPRED, FFAS3D. It gives a more robust consensus by combining multiple perspectives.
Does it just use sequence similarity for threading?
No, and this is important. It also uses predicted contact information derived from the deep learning steps we'll get to help guide the threading process. It tries to find templates that are not just sequence similar, but also structurally compatible with the predicted contacts.
Okay, and based on how well the threading works, like if it finds good matches or not, D-I-TASSER classifies the protein.
Yeah, into categories like trivial, easy, hard, or very hard, which they then simplify for practical purposes into just easy meaning good templates were found, and hard meaning templates were sparse or didn't align well.
And why classify them?
Because it influences how the rest of the pipeline runs. For an easy target, it might rely more on the template information. For a hard target, it will lean much more heavily on the de novo predictions from deep learning. It adapts the strategy.
Got it. So we have the MSA, potential templates, and a difficulty classification. Now comes the core deep learning bit, predicting spatial restraints.
Yes. This is where tools like deep potential and attention potential come in. Spatial restraints are predictions about the geometry of the final structure.
Like, these two residues should be X angstroms apart, or this bit should form a helix.
Pretty much. They predict several types. First, contacts. Which pairs of residues are likely to be physically close, say within 8 angstroms in the folded structure, even if they're far apart in the sequence? That coevolution signal from the MSA is key here.
Okay, contacts, what else?
More specific distances, not just close, but predicting the probability that the distance between two specific atoms, like the C-alpha or C-beta carbons, falls within certain ranges, like under 10 angstroms or between 13 and 16 angstroms.
More precise geometric information.
Exactly. And also crucial for getting the local structure right, predicting hydrogen bonds. These are specific interactions that stabilize secondary structures, like alpha helices and beta sheets. Predicting where these are likely to form helps lock down those fundamental building blocks.
And the deep learning networks figure all this out from the MSA features.
Yeah, using complex architectures, the sources mention things like residual networks, convolutional layers, even components inspired by AlphaFold 2's evoformer blocks. They learn the complex correlations between sequence patterns and 3D structure.
With multiple predictors running, how do they choose which predicted restraints to trust?
They use confidence scores. For contacts and distances, they select a certain number of the highest confidence predictions based on predefined cutoffs and probability distributions. The number they select might also depend on whether the target was classified as easy or hard.
Okay, so they select the most reliable predictions. Then what? How do these predictions actually guide the folding?
They're converted into energy potentials. It's a bit like turning each prediction into a tiny spring or magnet in a simulation. If the simulation tries to build a structure that violates a high-confidence prediction, say, it pulls two residues far apart that were predicted to be close, that structure gets an energy penalty.
So the simulation is constantly being nudged towards shapes that satisfy these predicted restraints.
Exactly. These deep learning restraints become powerful guiding forces within the simulation.
Which brings us to step 4, actually building the 3D model using folding simulations.
Right. This is where the more traditional physics-based simulation techniques come back into play, but now supercharged by the AI predictions. D-I-TASSER uses a really comprehensive energy function, or force field.
Not just the new deep learning potentials.
No, it combines those with dozens of other energy terms that come from classical ITASSER, representing things like bond lengths, angles, van der Waals. Forces our physical understanding of molecular interactions. It's a very rich force field.
And what simulation methods does it use?
Techniques like simulated annealing, or more commonly, replica exchange Monte Carlo, REMC. These are sophisticated ways to explore the vast energy landscape of possible protein conformations.
Trying to find the lowest point in that landscape, the most stable structure.
Exactly. Without getting permanently stuck in a less stable, incorrect fold. They usually start the simulations from initial structures based on the templates found by Lomitz 3, if any were good, and then let the system evolve under the guidance of that complex force field.
Are they simulating every single atom?
Usually not for the main search, that would be too slow. They often use a simplified model, like representing each amino acid mainly by its central carbon atom, the C-alpha, but ensuring the connections between them maintain realistic geometry.
Okay, that covers the core pipeline. But you mentioned multi-domain proteins were a special focus. How does D-I-TASSER tackle those specifically?
Right. This seems to be a key area where they aimed for improvement. The big challenge is getting the domains oriented correctly relative to each other.
So first they have to figure out where the domains even are in the sequence.
Yes. They use domain boundary prediction tools. Interestingly, they use a template-based one, Threadem, if the target was classified as easy, but a contact map-based one, FewPred, if it was hard, tailoring the tool to the situation.
Once they have the predicted domain sequences, what happens?
This is the clever part. They essentially treat each predicted domain sequence as its own mini prediction problem. They run Deep MSA2, Lomitz 3, and the deep learning restraint prediction independently for each domain.
So you get really detailed structural information within each domain.
Precisely. But you still need to assemble the whole thing.
Yeah.
So they create a new combined MSA, they call it checkerboard style MSA, that merges information from the full-length sequence alignment and the individual domain alignment.
Okay, a combined view.
Then they predict a new set of full-chain spatial restraints using this combined MSA. These restraints are particularly focused on predicting contacts and distances between residues that fall into different domains.
The interdomain connections.
Exactly. Finally, they assemble the final structure using the high-quality individual domain models they generated, guided by these new full-chain restraints, especially the interdomain ones. They mention using a tool called DEMO for this final domain assembly step.
So it's a multi-level approach.
Yeah.
Get the domains right individually, then focus specifically on predicting how they connect.
Yeah, and the sources really emphasize this domain splitting and reassembly protocol as being crucial for their improved performance on these challenging multi-domain targets.
Okay, that's a really detailed breakdown of the pipeline. It sounds incredibly thorough. The big question then is how well does it actually perform? Did all this complexity pay off?
That's the crucial part, isn't it? Let's look at the metrics and benchmarks.
First, how do they measure success? You mentioned TM score before.
Right. TM score is the main one they use for global structure similarity. It ranges from 0 to 1. A score over 0.5 generally means you've got the overall fold correct, even if details differ. Closer to 1 means a near-perfect match to the experimental structure.
And RMSD.
RMSD measures the average distance between corresponding atoms after superimposing the structures. Lower is better. It's good for local accuracy, but less reliable for overall fold assessment, especially with flexible regions or large proteins. They also mention Z-scores, often used in CSP competitions to rank methods relative to each other.
Okay, so focusing on TM score mainly. What did the benchmarks show? Let's start with those hard single-domain proteins of the 500 where finding templates was tough.
On that set, D-I-TASSER achieved an average TM score of 0.870, which is really quite high for hard targets.
How does that compare to the older I-TASSER methods?
It's a huge leap. Classic I-TASSER, the one heavily reliant on templates, only managed an average TM score of 0.419 on the same set.
Wow, less than 0.5 on average, so it often failed to get the fold right.
Pretty much. D-I-TASSER's score is more than double that. And compared to C-I-TASSER, an intermediate version with some contact prediction, which scored 0.569.
D-I-TASSER is still about 53% higher on average. That's significant.
Highly statistically significant, according to the sources. It's not just a small bump.
What about the number of proteins folded correctly? TM score above 0.5.
D-I-TASSER got 480 out of the 500 correct. Classic I-TASSER only managed 145, and C-I-TASSER got 329. So D-I-TASSER is solving structures correctly much more consistently, over 3 times more often than the classic version on these hard cases.
Looking head-to-head, did D-I-TASSER usually win?
Almost always. It had a higher TM score than classic I-TASSER in 99% of the cases, and higher than C-I-TASSER in 98%. It's a clear improvement across the board for these difficult single domains.
Okay, impressive gains there. What about the multi-domain proteins? That's where they put a lot of effort into the protocol. How did it compare to, say, AlphaFold 2?
They benchmarked it on 230 multi-domain proteins against AlphaFold 2. For the full-chain prediction, the entire protein structure, D-I-TASSER's average TM score, was 0.720.
And AlphaFold 2?
AlphaFold 2 scored 0.638 on average for the full chains in that set. So D-I-TASSER was about 12.9% higher on average for the complete multi-domain structure.
That's a noticeable difference. What about the individual domains within those proteins?
There, the gap was much smaller. Both methods did very well on the individual domains. D-I-TASSER averaged 0.858. AlphaFold 2 averaged 0.835. Still a slight edge for D-I-TASSER, but much closer.
So the main advantage seems to be in putting the pieces together correctly, the overall multi-domain assembly.
That's exactly what the sources conclude. The significant difference is at the full chain level, suggesting D-I-TASSER's advantage lies primarily in modeling the interdomain orientation accurately, likely thanks to that specific splitting and reassembly protocol we discussed.
They had an example, didn't they? That human protein, 6-ARDC.
Yes, a really illustrative one. AlphaFold 2 predicted the individual domains beautifully, high TM scores for each part.
What?
But it got the orientation between the domains completely wrong. The final full-chain model had a poor TM score, only 0.503, because the domains were just jammed together incorrectly relative to the real structure.
And D-I-TASSER?
D-I-TASSER's model had a much better arrangement of the domains, leading to a significantly higher full-chain TM score. The distance maps they show are revealing AlphaFold 2's predictions for distances between domains were often inaccurate, while D-I-TASSER's interdomain restraint predictions seemed more reliable, guiding a better assembly.
Okay, so benchmarks show promise, especially for multi-domain orientation. What about the big competition, CASP? That had a fare there.
CSP is the community-wide blind test, the critical assessment of structure prediction. D-I-TASSER participated in CIC15 under the name UM-TBM.
And the results.
Quite striking, actually. According to the assessment data presented in the sources, the D-I-TASSER server outperformed all other automated server groups.
In all categories.
In both the regular modeling category, mostly single domains, and crucially, in the interdomain modeling category focused on multi-domain assembly.
How big was the difference? They use z-scores in CSP, right?
Yes. For regular domains, D-I-TASSER's cumulative z-score was about twice as high as the standard public AlphaFold 2 server, MBIS-AF2 standard.
Okay, a solid win. But for interdomain modeling?
The difference was huge. D-I-TASSER's z-score was over 16 times higher than that public AlphaFold 2 server score for interdomain targets.
16 times. Wow. That really emphasizes the strength of their multi-domain approach in a competitive setting.
Absolutely. But here's something even more remarkable the sources highlight. CASP also includes human groups teams who can use manual intervention, expert knowledge, combined methods.
Right, potentially the best possible predictions. How did the D-I-TASSER server compare to them?
For regular modeling, it was competitive among the very top groups, server or human. But for interdomain modeling, the D-I-TASSER automated server clearly outperformed everyone, including all the human expert groups.
The automated server beat the humans at predicting multi-domain assembly.
According to the CASP15 results presented, yes. Its z-score was over 40% higher than the second best group overall, which was a human group. That's a really significant finding about the power of their automated protocol for these complex structures.
That is pretty remarkable. Okay, beyond CASP, they also tested it on a large set of known human proteins.
Yeah, nearly 2,000 human proteins from the PDB.
Yeah.
Again, comparing D-I-TASSER to AlphaFold 2.
And the overall result?
D-I-TASSER had a slightly higher average TM score overall, 0.931 versus 0.916 for AlphaFold 2. Statistically significant, the sources note.
But you said for many of these, both methods do really well.
Exactly. The average scores are high because many proteins in the PDB are, relatively speaking, easier targets now.
So did they look specifically at the harder cases within that human set?
Yes. They isolated about 250 proteins where at least one of the methods struggled a bit, scored below TM 0.8 on this more challenging subset.
The difference was bigger.
Much more pronounced. D-I-TASSER's average TM score was 0.699, while AlphaFold 2's was 0.633, and D-I-TASSER produced a better model, higher TM score, in almost 80% of these more difficult human protein cases.
So it reinforces that theme. D-I-TASSER seems to have an edge, particularly when the going gets tough, especially with multi-domain arrangements.
That seems to be the consistent message from the performance data presented.
One last practical thing on performance estimating model quality.
Yeah.
If you don't know the real structure, how reliable is D-I-TASSER's own quality estimate, the ETM score?
Right, the estimated TM score. It's calculated based on internal factors from the prediction process.
Okay.
How good the templates were, Lumet's 3 scores, how well the final model satisfies the predicted restraints, D potential scores, how stable the simulation was, and also incorporating AlphaFold's PLDDT score.
And does it actually correlate well with the true TM score?
Reasonably well, according to the sources. They report a Pearson correlation coefficient of 0.79 between the ETM score and the true TM score across their benchmarks.
So it gives you a decent idea?
Yeah, it's not perfect, but it's useful. It can help distinguish models that are likely correct, say ETM 0.5, from those that are probably wrong, with reasonable accuracy. It's a helpful guide for users interpreting the results for unknown proteins.
Okay, so the structure prediction itself looks very strong, especially on harder targets. But knowing the structure is often just a means to an end, right? The real goal is function.
Absolutely. What does the protein do? And the sources show D-I-TASSER is tightly integrated with a function prediction tool called cofactor.
Makes sense. Structure dictates function. So a good structure prediction should enable better function prediction. How does cofactor work?
It uses 3 main approaches, or pipelines, to predict different aspects of function. It predicts gene ontology, GO terms.
Judo terms, those describe biological process, cellular location, molecular activity.
Exactly. It also predicts enzyme commission, EC numbers, if the protein is likely an enzyme. And, very importantly, it predicts ligand binding sites, LBSs, where small molecules might bind.
How does it predict these? What are the pipelines?
The first is structure-based. It takes the 3D structure predicted by D-I-TASSER and compares it, using structural alignment tools, against a library of known protein structures, BioLip, that have experimentally determined functions and binding sites.
So if your predicted structure looks like a known kinase, it probably functions as a kinase.
Precisely. Structural similarity often implies functional similarity. The second pipeline is sequence-based. This uses more traditional BLAST-like searches to find proteins with similar amino acid sequences in annotated databases. Function can be transferred from close sequence homologs.
Less reliable than structure for distant relatives, maybe.
Generally, yes. And the third pipeline is PTI-based, using protein interaction data. It looks at which known proteins your query protein is known to interact with.
The idea being you are known by the company you keep. Interacting proteins often share pathways or cellular locations.
Exactly. Function can be inferred from interaction partners.
What about predicting the ligand binding sites,
Yeah.
where molecules actually dock onto the protein? That sounds critical, but maybe difficult?
Yeah, they detail a pretty sophisticated three-step process for LBS prediction. First, like the structure-based pipeline, it finds structurally similar proteins in the BioLip library that are known to bind specific ligands. It maps the location of those known binding sites onto the D-I-TASSER model.
So, an initial guess based on similar structures.
Right. Step 2, it takes the actual 3D coordinates of the ligands bound to those template structures and tries to physically place them, or dock them, into the predicted site on the D-I-TASSER model. It uses simulations, like Metropolis Monte Carlo, to refine the ligand's position and orientation to find a good fit.
Trying to find the best pose for the ligand in the predicted pocket.
Exactly. Step 3, since it might get slightly different ligand positions from different templates, it clusters all these docked ligand poses. Consistent clusters suggest a high-confidence binding site. It picks the most populated and confident cluster as the final predicted LBS.
They had a nice example of this too, didn't they? An enzyme.
Yes, acetyl-CoA acetyltransferase. D-I-TASSER predicted its structure almost perfectly, TM score 0.99. Then cofactor predicted where its substrate, acetyl-CoA, binds.
And how accurate was the binding site prediction?
Remarkably accurate. The predicted position of the acetyl-CoA molecule was extremely close to the real position in the experimental structure, RMSD of only 0.74 angstroms. And it correctly identified 22 out of the 23 amino acids known to line the binding pocket.
Wow, that really shows the power. Accurate structure leads to accurate prediction of molecular interactions.
It does. And they also applied this function prediction across the known human proteome.
What did they find there? Any interesting patterns?
They mapped the predicted functions back to the chromosomes. One interesting finding they highlighted was for chromosome 11. It showed a significant enrichment of functional annotations related to vision, things like visual perception, retin development, retinal ligand binding.
And is that consistent with what we know biologically?
Yes, apparently experimental studies have linked many genes on chromosome 11 to various ophthalmic diseases. So seeing that pattern emerge from the computational predictions is a nice validation and shows potential for uncovering functional connections.
Okay, so D-I-TASSER looks like a real advance, strong on structure, especially tricky ones, and linked to powerful function prediction. But it's not perfect, right? What are the remaining challenges the sources point out?
No, definitely not perfect yet. They're quite upfront about the limitations. Even with the improved multi-domain protocol, they say the performance there is still not entirely satisfactory. Meaning, predicting the precise distances and orientations between domains often still has lower accuracy compared to predicting the structure within a domain. There's still room for improvement in getting that full assembly just right.
What about proteins that work together in complexes, oligomers?
That's a major issue they flag. Proteins that form complexes where multiple chains come together, either copies of the same protein or different ones.
Why is that so hard for these methods?
It goes back to that coevolutionary signal from the MSA. The deep learning models use it to predict contacts. But in an oligomer, the signal doesn't distinguish between contacts within a single chain, interchain, and contacts between different chains, interchain.
So the network might predict two residues are close, but it doesn't know if they're close on the same molecule or because two molecules are touching at that point.
Exactly. The sources show an example, T1070-day D1, where the individual chains are modeled okay, but the prediction of how they fit together in the complex is poor because the interchain contacts weren't accurately distinguished or predicted. This is a big limitation for understanding many biological machines.
Are there other limitations?
Computational resources are mentioned. The deep learning parts, like deep potential, were trained on GPUs with relatively limited memory, 10 GPUs mentioned.
Which means
Which means they couldn't necessarily use the absolute largest, deepest neural network architectures, or incorporate every potentially useful input feature, more computing power could potentially lead to further accuracy gains.
And getting the input data, the MSAs.
That's becoming a bottleneck, too. Searching the ever-growing metagenomic databases takes significant time and resources.
And what if you just can't find many related sequences?
That's the shallow MSA problem. For some proteins, maybe from rapidly evolving viruses or just unique proteins, you can't build a deep, diverse alignment. And methods like D-I-TASSER, which rely heavily on that coevolutionary signal, struggle when the MSA is shallow. Predicting structures for these orphan proteins remains tough.
And the ultimate challenge.
Predicting the structure of protein-protein complexes, how different protein molecules dock and interact. The sources explicitly state this is a major problem that still lacks an effective general solution. D-I-TASSER focuses on single chains, which might have multiple domains, but predicting the interfaces between separate interacting proteins is the next big frontier.
Okay, so significant progress, but still some major mountains to climb. Where do they see things going? What's the future direction?
They seem optimistic about the hybrid approach itself. That combination of deep learning, pattern recognition, and physics-based simulation refinement seems really powerful.
So building on that success.
Yeah. They suggest extending the D-I-TASSER protocol to specifically tackle some of these challenges, like developing better strategies for orphan proteins with shallow MSAs.
And the big one, protein complexes.
That's mentioned as a key target for future work, extending this hybrid philosophy to predict the structures of protein-protein interactions. And of course, leveraging more computational resources could enable more complex models and potentially higher accuracy across the board.
It feels like while deep learning provided this massive leap, the integration with simulation, like in D-I-TASSER, is key to refining those predictions and tackling the really hard cases, especially multi-domain assembly.
That seems to be the takeaway. It's not just AI replacing everything, but AI providing incredibly powerful information to guide and enhance simulation methods that are rooted in our understanding of physics.
Okay, let's try to quickly recap the key takeaways from this deep dive into D-I-TASSER.
All right. First off, it's a hybrid approach, right? It cleverly blends deep learning predictions with traditional physics-based folding simulations.
Second, it starts strong with sophisticated data gathering deep MSA2 for mining sequence databases, including metagenomics and LOMITS3 for finding structural templates.
Third, the core deep learning part predicts really detailed spatial restraints, contacts, distances, even hydrogen bonds, which act as crucial guides during the simulation phase.
Fourth, this seems really key, it has that specialized protocol for multi-domain proteins, splitting them, modeling domains individually, and then carefully reassembling them using specifically predicted interdomain restraints.
Fifth, the performance data looks strong. Significant improvements over older I-TASSER versions and notable advantages compared to AlphaFold 2, especially on harder targets and, very strikingly, in the CASP15 results for multi-domain protein assembly.
Sixth, it connects structure to function through cofactor, using the predicted shape, sequence homology, and interaction networks to make predictions about jugo terms, enzyme activity, and ligand binding sites.
And finally, despite the progress, major challenges are still out there. Getting multi-domain and oligomer structures perfect, handling proteins with limited sequence data, shallow MSAs, computational scaling, and the grand challenge of predicting protein-protein complex structures accurately.
The sources seem to end on a hopeful note, though, suggesting this hybrid strategy holds real promise for tackling those remaining frontiers, like orphan proteins and protein complexes.
Yeah, it leads to that provocative thought, doesn't it? Could this continued fusion of advanced AI learning complex patterns from data with sophisticated physical simulations be the path forward? Could it finally allow us to reliably predict the structure of almost any biological machine, opening doors to understanding and engineering biology in ways we can barely even imagine today?
It certainly feels like we're getting closer to answering some of biology's biggest questions.
Exciting times in structural biology, for sure.
Thank you for joining again.